We generally accept that human evolution was a slow, steady march over millions of years. But when you look closely at our DNA, the fossil record's gradual timeline begins to show significant disruptions. We carry abrupt structural changes in our genetic code that standard evolutionary models have difficulty explaining through slow mutation. Take our closest primate relatives. Chimpanzees, gorillas, orangutans, and bonobos all have 48 chromosomes. We have exactly 46. This chart shows the genomes of great apes alongside humans. The discrepancy exists because two distinct ancestral primate chromosomes fused end-to-end to create our human chromosome 2. We can see the evidence of this event in the vestigial telomere sequence located right in the middle of the chromosome, marking the exact point where the two ends joined. Chromosomal fusions of this magnitude are rare in mammals, often resulting in reduced fertility or unviable hybrids. For this specific fusion to spread through and define an entire population, it would have required either a very small founding group or an intense selection pressure that effectively favored this new 46 chromosome structure over the ancestral 48. Beyond the structural fusion of the chromosomes, the actual genetic code contains a separate anomaly. We have 3,171 regions in our DNA known as human accelerated regions, or AHRs. For nearly 300 million years, across fish, reptiles, and our primate ancestors, sequences like AHR1 remained biologically locked and unchanged. Within the human lineage, this ancient, stable sequence broke its 300 million year stasis. AHR1 rapidly accumulated 18 mutations in a geologically short window. Researchers have now mapped over 3,000 of these regions that underwent rapid rewriting. When you look at what these specific genes control, they cluster around the development of the human cortex, language organization, and the neural networks responsible for abstract thought. While natural selection provides a framework for gradual change, the standard evolutionary model faces challenges explaining the speed of these specific upgrades. The acceleration in these conserved brain development sequences happens significantly faster than random mutation typically accounts for. In 1700 BC, a text called the Atrahasis Epic was recorded, drawing on Sumerian sources that predate it by centuries. Most ancient creation myths describe gods breathing life into dust or forming humans through divine will alone. The Sumerian account describes a biological procedure. It records two beings, Enki and Ninhursag, taking the genetic material of an existing hominid and combining it with their own essence to produce a hybrid. The oldest records we possess describe a procedural genetic intervention that aligns with the biological mechanisms we are only now identifying in our own DNA. The texts do not describe a single moment of magical success, but a rigorous process of trial and error. They list specific defects of the early prototypes, describing beings with missing functional organs, incorrect physical proportions, and an inability to procreate. These ancient descriptions correspond to the types of complications modern geneticists encounter when species are hybridized. The texts describe the biological hurdles, such as barrenness and organ failure, that characterize failed hybridization outcomes. According to the record, the process continued until the creators stabilized a version that was capable of surviving independently and procreating. This ancient account of creation places the intervention within the same general evolutionary window between 300,000 and 700,000 years ago, when our DNA was undergoing its most rapid and unexplained cognitive rewriting. The Sumerian texts are explicit about the motive. Humans were not created for companionship, but to serve as a biological workforce to manage resources after a group of lesser gods rebelled against their labor. This workforce required specific cognitive traits, the ability to follow complex instructions, use sophisticated tools, and coordinate labor while remaining manageable. This requirement for abstract thought matches the archaeological record. Approximately 50,000 years ago, we see an event often called the cognitive revolution, the sudden arrival of complex symbolic art and language. We see a transition from basic survival tools to advanced cultural artifacts, like these flutes with precision-carved holes, proving an abrupt capability to craft instruments for musical expression. Anatomically modern humans survived for nearly 200,000 years using the basic toolkits and fire-making skills inherited from their predecessors. Then, in a geologically short window, we expressed the full suite of complex cognitive capabilities the Sumerians say we were engineered to possess. This leaves a timing gap. If the brain-enhancing HAR mutations were established over 300,000 years ago, what caused those capabilities to remain dormant until the explosion of culture 50,000 years ago? The Sumerian account describes Enki as a creator who eventually went rogue, acting as an advocate for humanity by secretly gifting them advanced knowledge against the wishes of the other Anunnaki. This gift of knowledge could represent a secondary intervention, a cascading trigger that finally activated the advanced cognitive hardware that had been genetically present but behaviorally latent for thousands of generations. While we cannot scientifically prove ancient engineering, the alignment between our unresolved genetic anomalies and the oldest recorded procedural text on Earth makes the idea of humanity as a biological experiment difficult to ignore.